Hi, good evening everyone. I Dr. Dipankar Ghosh, Assistant Professor Microbial Engineering and Algal Biotechnology Laboratory, Department of Biotechnology, JS University, Kolkata. Today I am going to discuss and share some of my knowledges on structure-based design for screening and repurposing antivirals against COVID-19. Coronaviruses are the large family of viruses which may cause illness in animals or humans. In humans, several coronaviruses are known to cause respiratory infections ranging from common cold to more severe diseases such as Middle East Respiratory Syndrome (MERS) and acute severe respiratory syndrome (SARS), and the most recently discovered coronavirus causes coronavirus disease COVID-19. COVID-19 is the infectious disease caused by the most recently discovered novel coronavirus. The new virus and the disease were unknown before the outbreak began in Wuhan, China in December 2019. COVID-19 is now a pandemic which affecting many countries globally. This is a typical example of a novel coronavirus which causes COVID-19 disease. Mostly it contains nucleocapsid protein which actually surrounds the positive strand, single strand RNA molecule, which is surrounded by different envelope glycoprotein E, spike protein S, membrane glycoprotein M, and lipid bilayer. Phylogenetic tree showing the relationship of Wuhan denoted as red to selected coronavirus is based on nucleotide sequences of the complete genome. The viruses are grouped into 4 genera. The prototype shown: the alpha coronavirus sky color, beta coronavirus pink color, gamma coronavirus green color, and delta coronavirus light blue color. The subgroup clusters are labeled as 1A and 1B for alpha coronavirus and 2A, 2B, 2C and 2D for the beta coronavirus. This tree is based on the published trees of Coronaviridae group and reconstructed with sequence of the complete RNA-dependent RNA polymerase coding regions of the representative novel coronaviruses using maximum likelihood method. Severe acute respiratory syndrome coronavirus SARS-CoV, SARS-related coronavirus SARS-CoV, and Middle East Respiratory Syndrome coronavirus MERS-CoV, porcine enteric diarrhea virus PEDV, Wuhan seafood market pneumonia, Wuhan HU1, bat-CoV RATG13 showed high sequence identity with SARS-CoV-2. Reservoir and mode of transmission of coronaviruses. Suspected reservoirs of the SARS-CoV-2 are red encircled. Only alpha and the beta coronavirus have the ability to infect human. The consumption of the infected animal as a source of food is the major cause of animal to human transmission of the virus, and due to close contact with an infected person, the virus is further transmitted to healthy person. Dotted dark arrow shows the possibility of viral transfer from the bat, whereas the solid black arrow represents the confirmed transfer. Next slide deals with the beta coronaviruses Genome Organization. The beta coronaviruses for human SARS-CoV-2 and SARS-CoV MERS-CoV genome comprises of 5' untranslated region, the 5' UTR, open reading frame ORF 1A and 1B green boxes representing here encoding non-structural proteins NSP for replication structural proteins including spike, which shows in the blue blocks, and the envelope, the maroon box, the membrane in the pink box, and the nucleocapsid cyan box proteins. Accessory proteins light gray boxes such as ORF3, 6, 7A, 7B, 8 and 9A in the SARS-CoV-2 novel coronavirus genome, and the 3' untranslated region, that is 3' UTR. The dotted underlined in the red are the protein which shows key variation between SARS-CoV-2 and SARS-CoV. The length of the NSP and ORFs are not drawn in scale. Slide typically deals with the life cycle of the novel coronavirus or the SARS-CoV-2. The life cycle of the SARS-CoV-2 in the host cell begins its life cycle when the S protein binds to the cellular receptor ACE2. After receptor binding, the conformation change in the S protein facilitates the viral envelope fusion with the cell membrane through the endosomal pathway. Then SARS-CoV-2 releases the RNA into the host cell. The genome RNA is translated into the viral replicase polyproteins PP1A and 1AB, which are then clipped into small products by the viral proteinases. The polymerases produces a series of subgenomic mRNAs by discontinuous transcription and finally translated into relevant viral proteins. The viral proteins and the genome RNA are subsequently assembled into virion in the endoplasmic reticulum and Golgi and then transported by the vesicles and released out of the cell. ACE2, so-called angiotensin converting enzyme 2, ER endoplasmic reticulum, ERGIC, the endoplasmic reticulum Golgi intermediate compartments. The next slide deals with the COVID-19 towards understanding of pathogenesis. The clinical course of the SARS-CoV-2 infection could be divided into three phases: the viremia phase, acute phase, pneumonia phase, so-called, and severe or the recovery phase. The patient with the competent immune functions and without obvious risk factor, that is old age, comorbidities, etc., may generate effective and adequate immune responses to suppress the virus in the first and the second phase without immune overreaction. In contrast, the patients with the immune dysfunction may have a higher risk of failing the initial phase and becoming a severe or the clinical type with higher mortality. Therefore, the treatment of COVID-19 should be based on the staging of patients, and the window of opportunity may lie between the first and the second phases, when clinician deterioration is observed with the evidence of the abrupt inflammation and hypercoagulable status. With no proved antiviral, early intervention has mainly focused on the correct timing of disease stages and implementing ways to stop or slow the disease progression. Once the patients enter the critical status, no magic bullet could be relied on other than comprehensive management. This hypothetical pathogenesis of COVID-19, it deals with the x-axis, where it refers to the days after SARS-CoV-2 infection. The disease course is divided into three phases, as I said earlier. The y-axis shows the trend of T cell, B cell, and inflammatory factors, D dimers, viral load in patients. In section A, the trend of each indicator in severe COVID-19 patients has been represented, whereas the B section deals with the trend of each indicator in severe COVID-19 patients after low molecular weight heparin, LWMH, and high-dose intravenous immunoglobulin IVIG therapy. The shaded areas represent the recommended intervention time for LMWH and IVIG treatments. Viral and the host factors that influence the pathogenesis of SARS-CoV-2. Bats are the reservoir of a wide variety of coronaviruses, including severe acute respiratory syndrome coronavirus SARS-CoV-like viruses, and SARS-CoV-2 may originate from bats or unknown intermediate hosts and cross the species barrier into human. Virus-host interactions affect the viral entry and replication. Upper panel, the viral factors. SARS-CoV-2 is an envelope-positive single-stranded RNA coronavirus. Two-thirds of the viral RNA mainly located in the first open reading frame ORF1A and 1B, as I said earlier, encodes 16 non-structural proteins, NSPs. Rest part of the virus genome encodes 4 essential structural proteins, including spike glycoprotein, small envelope E protein, matrix M protein, and nucleocapsid N protein, and also several accessory proteins like ACE glycoprotein of SARS-CoV-2 binds to the host cell receptor angiotensin converting enzyme ACE2. That is a critical step for viral entry. The possible molecules facilitated membrane invaginations of the SARS-CoV-2 endocytosis, and still it's unclear. The other virus protein may contribute to pathogenesis. The host factor, lower panel, can also influence the susceptibility of the infection and disease progression. The elderly and people with underlying disease are susceptible to SARS-CoV-2 and tend to develop into critical conditions, where the RBD, representing the receptor binding domain, HR1 is the haptad repeats and HR2 is the haptad repeat 2. Now this is a nice way of summary of pathogenesis by SARS-CoV-2, where it says how SARS-CoV-2 enter and replicate inside the host, how they accelerate the antigen presentations and induces the cellular immunity and humoral immune responses. Now coming into the main scenario, then what is the driving force for the structure-based design for screening and repurposing antiviral against COVID-19? Why these repurposing and structure-based designs are required for combat with the COVID-19 pandemic situation? The causative agent for COVID-19 is SARS-CoV-2, the severe acute respiratory syndrome coronavirus 2. Other similar agent previously known as Middle East respiratory syndrome MERS, virus MERS-CoV, and MERS and the SARS-CoV. They attack patients' lower respiratory system by invading the pulmonary epithelial cells, delivering their nucleocapsid and hijacking the cellular machineries to replicate in the cytoplasm. The virus family also affects the heart, kidney, liver, gastrointestinal system, and central nervous system. SARS-CoV-2 belongs to the Coronaviridae family of enveloped single-stranded positive strand ribonucleic acid RNA. Structure. The structures of the SARS-CoV-2 is in close resemblance to that of SARS-CoV. The SARS family contains 14 binding residues, out of which 8 amino acids are specifically conserved for the SARS-CoV-2. Importantly, the binding residues of this family interact with ACE2, the angiotensin converting enzyme 2, directly. Since the quick transmission of the coronavirus can be catastrophic for the entire world, the healthcare authorities have suggested certain preventive measures. The quarantine, the infected patients, aggressive testing, rapid diagnosis of suspected victim, use of appropriate masks, frequent hand washing will help to encounter the control of progression of this severe disease. But unfortunately, currently no drug or vaccine is available for coping this disease. Moreover, SARS-CoV-2 so far more contagious compared to other flu viruses, as the pre-symptomatic or asymptomatic person is capable to infect more than 2 healthy individuals. The researchers are now focusing on repurpose strategy of existing drug, and this is the major driving force why we should follow up or continue the structure-based repurposing to find out or fetch out the antivirals against the COVID-19. It is good to know that the scientists are working in this field have suggested the uses of some known broad-spectrum antiviral drugs like nucleoside analogs, HIV protease inhibitors as promising treatment methodologies. The RNA-dependent RNA polymerase, the RDPR or the RDRP, the angiotensin converting enzyme to ACE2 are also viable drug target for the COVID-19 treatment at present scenario. Some antiviral drugs like favipiravir, ritonavir, oseltamivir, lopinavir, granciclovir, remdesivir are clinically tested against the COVID-19 infections. The chloroquine, an antimalarial drug, has also been proven to be effective in treatment towards COVID-19. Before entering into the different segments of the structure-based design for screening antiviral compounds and also antiviral repurposing, we need to know what is structure-based drug design. The structure-based drug design is the design and optimization of a chemical structure with the goal of identifying a compound which is suitable for the clinical testing and that is a drug candidature. Secondly, what is the drug repurposing? The drug repurposing involves the investigation of an existing drug for new therapeutic purposes. Give an example, likely FDA approved drugs candidates which are normally being used for the SARS, MERS, Ebola, HIV and malaria can also be applied to test their efficacy against the SARS-CoV-2 or the novel coronavirus. First and most promising approach is the protein interaction map, which reveals the target for the drug repurposing against the SARS-CoV-2. So it consists of 2 major important steps. Number 1, the affinity purification mass spectroscopy, so-called APMS, and it followed by the protein-protein interaction network maps generation. So this is an example of a typical experimental workflow for the APMS studies, where you have 27 bait which has been already chosen from the novel coronavirus surface proteins, and out of these 26 proteins and 1 is the variant. Then it has to be tagged with the stripped tagging, and then it has to be expressed in the HAK293T cells in a biological triplicate, and there has to be followed affinity purification to purify that particular kind of bait proteins, and then has to be analyzed through the mass spectroscopic analysis, and that will give you some PPI scoring, and this PPI scoring will help to generate the PPI or protein-protein interactome map generations. Next is the global analysis of the SARS-CoV-2 protein interactions. So overview of the global analysis performance shows that the gene ontology geo-enrichment analysis was performed on the human interacting proteins of each vital protein. The p-values calculated by the hypergeometric test and a false discovery rate was used to account for the multiple hypothesis testing methods. The top geoterm of each viral protein was selected for the visualization. Now, in a simpler way, what it tells? It actually tells the different annotated nucleotide or the protein sequences which are present in the novel coronavirus, how they are related or how they are interacting with the different molecular network in a host like human. So it may be related with the nuclear transport, or it can be involved in the cellular component disassembly, can be involved in the gene silencing, microtubule based process, organelle inner membrane integration, or sometimes regulation of the reactive oxygen species generation, or lipoprotein metabolic processes, and so forth. And moreover, the SARS-CoV-2 genome annotation, which the color intensity is the proportional to the protein sequence similarity with the SARS-CoV homologue, and when it's only possible when the homologues exist in the nature. The next segment deals with the SARS-CoV-2 protein-protein interaction network or near reactome generations. So SARS-CoV-2 protein-protein interaction network says 332 high confidence interactions between 26 SARS-CoV-2 protein, which are represented as a red diamond, and the human protein circles drug target orange, protein complexes yellow, and protein in the same biological purposes are blue in color. Each color proportional to the MEST score, the edge thickness proportional to the spectral count. The physical interactions among the host protein, which represented as the thin black lines, were curated from the quorum or INT-ACT and the reactome softwares. These are publicly available as a web-based service approach. So an interactive PPI map can be found. I mentioned especially the UCSF lab is providing this particular web server where you can actually able to generate this PPI map if you have a complete genome sequence of particular kind of novel viruses. In our case, it is novel CoV-2, and you can also do biological replication based on the independent samples. So in more simpler way, what it tells about that? So it tells about how different surface proteins or different other proteins, non-structural, structural or accessory proteins, how and where they are actually interacting, and what are the different pathways which are involved, and where this protein can also get target. Like, give an example, NSP1. In the reactome analysis, PPI reactome analysis shows that the NSP1 can have a high interactivity with DNA polymerase alpha subunit. The similar way, the NSP4 having high interaction between the trans intermembrane helix complex. On the other hand, NSP6 or NSP5, NSP5, C145A can also interacting with the iron transport channel. Similar way, NSP9 having higher affinity towards the nuclear pore interactions, and so forth. This is most interesting, like how different NSP proteins are reacting, or they are interacting, or they are supposed to be interacting with different. It's kind of prediction. It has to be verified by validation experimentations. Now, PPI-based reactome analysis not only tells about like what are the proteins, whether it's structural, unstructural or accessory protein, how they are not only related with different pathways present on the host, but it can also tells you how they are or what they are the most prominent or the closed hypothetical drug targets. Now, in our case, it has been shown that the drug human target network shows the PPI of the SARS-CoV-2 bait with approved drug, which are represented as green, clinical candidates represented as yellow, and preclinical candidate represents as a purple color with experimental activities against the host protein. The host protein which are represented as the white background, or previously known for some other host factor which represented as the gray background, are shown over here. Now, the specific reactome analysis using the PPI network, it gives you some hypothetical idea or some kind of information, like it can be useful, and you can validate by do experimental analysis. So it says here in a simpler way how different structural and non-structural accessory protein could be a target of different kind of drug candidates which are never being used before to reacting with those kind of proteins to eradicate or minimize the efficacy of the COVID-19 novel coronavirus. Now, it says, give an example, So if you choose NSP1, it can be treated or it can be reacting highly with the rapamycin. Similar way, the valproic acid can be also used to interact with the NSP4. So NSP4 should be the target of that particular kind of drug molecule. In a similar manner or similar fashion, chloramphenicol or the tigecycline or linezolid, they can also be used to target NSP8, or NSP14 can also be targeted by using the ribavirin, never being used before to treat like that. Similar way, you can also use the rapamycin to target the NSP or the N or E envelope proteins or the nucleocapsid proteins. You can use rapamycin, you can use selenoxin. So in some few cases you can also use midostaurin to interact with the ORF10. ORF9B can also be reacted with the ruxolitinib, and ORF8 can also be reacting with rapamycin. So these are all hypothetical. It has to be tested, it has to be clinically tested and try and make sure like whether this target ORFs or envelope or the membrane proteins can be really reacting with this kind of antiviral compounds or not. And then if it works, then it's really a nice story to somehow minimize, or it can kill the spread, or it can kill the efficacy of the COVID-19. Structure-based design for screening of antivirals. To determine the antiviral compounds which are novel or pre-existing after repurposing, it is very important to know how interactive they are and how efficiently they interacting with the target molecules in the virus as well as on the human. So to do that, especially the structure-based design for screening of antiviral, it requires certain informations. Like it requires information about the SARS-CoV-2 genome sequence, complete information, especially considering the structural, non-structural and the accessory protein information, their sequences. And then it requires also the information about the targeted viral protein which are present on the surface, especially which are actually responsible for the pathogenicity or for the recognition of the human host receptors. Finally, all this information will put together and assemble into a structure-based virtual screening process. And this process also requires certain insight of viral protein structural informations. Now, altogether, they will give you a very nice workflow that will help to identify the new antiviral compound against COVID-19. Let's discuss about the detailed workflow, how it could be worked. That means the structure-based design workflow, how it will be working to screen different antiviral compounds against COVID-19. Firstly, the genome sequence of the COVID-19 has already been deposited along with the different variant sequence information in the gene bank repository or in the big data set. The application of the bioinformatics is useful to find out the genome annotation by using the basic local alignment search tool, so-called BLAST, which is a sequence alignment program. Finding out the surface receptor which are present on its surface and what their sequence is. The development of the protein model or the three-dimensional structure using the homology modeling, as not all the spike or the surface proteins are actually crystallized form available in the RCB database. That's why we need to somehow follow the homology modeling using a reference point of the SARS-CoV-1. Now finally, the application of the molecular docking and MD simulation will be useful for the discovery and validation of the novel drug in the in silico approaches. The important first segment is the gene annotation by the BLAST for the identification of coding regions of the genome, which are so-called exons. The possible analysis to annotate gene can be done by similarity searches, gene cluster prediction for secondary metabolites, identification of the transmembrane domain in the protein sequences, finding gene ontology terms and the pathway informations. Second most important segment is the developing protein model from the protein database using homology modeling. Homology modeling is basically modeling of the 3D structure of a protein by exploiting structural information from the known configuration of similar kind. Like in case of SARS-CoV-19, the similar virus is the SARS-CoV-1. So it will include a few steps, likely fetching the target protein sequence of SARS-CoV-2, the template identification where a protein template whose 3D coordinates are known and shares the maximum similarities with our target sequence will be selected. Similarity check between the target and the template can be done using BLAST. Finally, the model will be generated. It will include the backbone, loop and sidechain generation, model optimizations, MD simulation to develop the trajectories, and finally it will go for the validation experimentations. In the study, we have included an example or demonstration how it will work, the molecular docking. So to that you need to have a target selection, and you should have some information about the ligand selection database. Now you should start docking using the SwissDock, which is a web-based server, and then SwissDock is based on the docking software EADock DSS, whose algorithm consists of the following steps. The many binding modes are generated either in a box or local docking, or in the vicinity of all target cavities, which are so-called the blind docking. Simultaneously, their charm energy are estimated on a grid basis. The binding models with the most favorable energy in terms of grief-free energy and evaluated with the FACT software and clustered. The most favorable cluster can be visualized online and downloaded on your computer. Once the job has already been terminated, you will receive an email with the link to reference complex and predicted binding mode. They can be converted into a favorable format or used directly. The ViewDoc, which is a plugin of UCSF Chimera, is very convenient to explore the predicted binding modes. For experimented users, the charm PSF or CRD or RTF or PAR files can be generated or provided by the servers after the energy calculations, and it will give you a complete idea about how they are closely related with the protein based on the X-ray binding mode in a ball stick or different other modes. So finally, you will come up with the binding affinity plot. There are different clusters will be available. You can have a idea about what are the interactive binding affinity, and you can also rotate the structure which are available in the SwissDock results section. For the structure-based design of screening, we have chosen the chloroquine, a novel drug which is anti-malarial drug previously being used. So we use SwissDock, or the demonstration we have given earlier, to dock the chloroquine or novel drug with the COVID-19 novel surface protein 3I6G, 6VXX and C6W02. These are the different crystal structure or the homology based model which are developed from the RCB database, and now we put together and dock to each other and find how chloroquine binds with the COVID-19 spike protein 3I6G protein, and it will also give you the binding mode where it binds, which amino acids are responsible for binding, and it will also give you some idea about the free energy change which correspond to the binding affinity in terms of negative value or free energy change towards the entropically favorable state. Based on the previously mentioned workflow, a stepwise structure-based virtual screening pipeline was adopted, considering the putative binding site of the SARS-CoV-2 M-Pro protease, NSP12 polymerase, NSP13 helicase to identify the potential hits. The identified potential hits against each target were subjected to in silico ADMET prediction analysis, which includes the absorption, distribution, metabolism, elimination and toxicity, to clarify the dark likeliness and the toxicity potential towards the potential host. The current slide has clearly been depicted, and it has been shown that the SARS-CoV-2 3CL protease structure and how they are interacting with CMP, that is chlorpheniramine compound, and how the different domains of this protease interacting with the CMP drug candidate, and which amino acids are also involved and interacting in the specific catalytic domain or the motif responsible. On the section B on the same slide has shown that the binding affinity with different kind of CMP target drug molecules, and it shows different CMP molecules having different molecular structure, and based on this molecular interaction with the SARS-CoV-2 3CL protease giving different kind of free energy change. So based on the free energy change towards the negativity, it shows that the efficiency or efficacy of the interactability. In a similar fashion, NSP12 polymerase and NSP13 helicase has been docked with the different CMP varieties, and it shows how they're interacting with each other in terms of free energy change, and that will determine how efficient the CMP class of molecules against this NSP12 polymerase and NSP13 polymerase drug targets. In another study, the similar SARS-CoV-2 M-Pro protease is complex or docked with the 11A and 11B two very unique antiviral candidates. The slide has been clearly shown that how they're interacting and what are the amino acids which are responsible for binding with 11A and 11B. So these are the different pictorial presentation. And finally, in an in vitro inhibition study of the viral main protease MPRO inhibitors against the SARS-CoV-2 has been depicted. Here, the Vero E6 cells were treated with a series of concentration of indicated compounds of 11A and 11B and infected with the SARS-CoV-2 at a MOI or multiplicity of infection of 0.05. At 24 hours post-infection, the viral yield is the cell supernatant was quantified by the plaque assay. The cytotoxicity of these compounds in the Vero E6 cells was also determined by using the CCK8 assay. The left and right y-axis of the graphs represent the percentage of inhibition of the virus yield and percentage of cytotoxicity of the drugs, respectively. The viral RNA copy numbers in the cell supernatant were also quantified during this experiment by quantitative reverse transcriptase PCR. In another study where the ACE2 and TMPRSS2 protease has also been targeted to the diverse ranges of protease inhibitors, but experimentally proven stuffs or the information available, and it has been evident that the protease inhibitors which are used against the TRMPRSS2 are based on the neutralization efficacy. The efficacy was very low for the SARS-CoV-2 compared to the SARS-CoV-1. Finally, not the least, the top 20 drug which are approved shows the highest stocking score using this aforementioned structure-based design computational workflow. So in this tabular presentation, it has been shown that the 20 top drugs which are casually or normally used for the different other clinical purposes, likely to treat the antivirus, hepatitis V, for vitamins, sometimes they use for the cardiovascular diseases, sometimes they are treated for the cancer disease, and also for NSAID cure, or sometimes they use as an anti-convulsant as well. So now these 20 drugs, now they are targeted to use for the reactivity with the CoV-2, which is SARS-CoV-2 COVID-19. And the below, these are the different interactions has been shown. So I have just chosen two different interactions between the CoV-2 with ribavirin and the telbivudins. So as a take-home message, it can be summarized that the current computational workflow of structure-based design for screening and repurposing antivirals against COVID-19 is an accelerated bioinformatics approach towards identification of novel known and unknown surface receptors or the target constituents of the COVID-19, which will definitely help to screen and identify new novel drugs or existing drugs through purposing against the COVID-19. Secondly, it will also minimize the time of effective drug discovery with the conventional approaches, which will indirectly torch a new light to combat with the deadly COVID-19 pandemic situation. Thus far, the molecular docking and virtual screening and other computational techniques are widely used methods to understand the phylogenetics, molecular aspects of the protein-ligand interactions during the drug discovery processes. Virtual screening has also been used in drug discovery against emerging and fatal diseases including SARS-CoV proteases, hepatitis C virus, RNA-dependent RNA polymerase, dengue viruses, and Ebola viruses. Therefore, in this slide, I would like to thank WHO, World Health Organization, for choosing me for a very informative and quality training program for enlightening my knowledge on COVID-19 to spread general awareness towards general audiences. Finally, I would like to acknowledge JIS Group and educational initiatives, especially JIS University Kolkata, for providing me this opportunity to deliver the talk through the webinar series. Secondly, I'd like to acknowledge my funding agency, TJM Startup Grant, for conducting research. And finally, I am very much grateful to my PhD scholars and my MSc research scholars in Microbial Engineering and Algal Biotechnology Laboratory, JIS University, Kolkata. Thank you very much for listening, for your attention. Great. Thank you.